All right, sorry, my computer just stopped recording. But so a photosystem, like I was saying, a photosystem is made up of chlorophyll molecules, small organic molecules, and proteins. And so a photosystem consists of a reaction center complex surrounded by light harvesting complexes. And you'll see this a little bit later in some of the diagrams. But the light harvesting complexes transfer the energy of photons to the reaction center. So the reaction center complex, it has a special pair of chlorophyll A molecules that I will talk more about later. But this photosystems are in the thylakoid membrane, in the chloroplast, in the cell. All right, so a primary electron acceptor is in the reaction center, is the molecule capable of accepting electrons and becoming reduced. And so the special pair of chlorophyll A that I was talking about in the last slide transfers energy to this primary electron acceptor. So this is basically what I was just talking about, and it gives like a more up close version of these pictures. So I'm going to fast forward. So yeah, so if you start in the top left-hand corner, you see the photon coming in, and these little green things are the pigment molecules. And so the energy goes from pigment molecule to pigment molecule in the light harvesting complex, which is like the purple things. And then, so it's transferring energy from molecule to molecule until it reaches the special pair of chlorophyll A molecules, which is in the reaction center complex, which is the blue bit. And then that's when the electron gets excited and transfers to the primary electron acceptor, which is that blue box. This picture is weird and confusing, so I'm just going to go ahead and skip over it. All right, so there are two types of photosystems in the thylakoid membrane. So photosystem II, it's called PS2, it functions first. So it's a little, counterintuitive, but photosystem II functions first. And it's best at absorbing a wavelength of 680 nanometers. So we call the reaction center chlorophyll A of PS2, we call it P680. So photosystem I is best at absorbing a wavelength of 700 nanometers. So not surprisingly, the reaction center chlorophyll A of PS1 is called P700. These are both, like P680 and P700 are both in the red end of the visible light spectrum, but P700 is just further red, if that makes any sense. So P680 and P700 are almost identical, but they're just associated with different proteins in the thylakoid membrane. All right, so moving on to a slightly different kind of topic. Linear electron flow is the flow of electrons through the photosystems and other molecular components built into the thylakoid membrane. And that's the key to driving the synthesis of ATP and NADPH. So the linear electron flow occurs during the light reactions of photosynthesis. And so there are 8 steps on how it produces ATP and NADPH. So I'm going to go over them really quickly, as quickly as I can anyway. So the first step is a photon of light hits a pigment in a light harvesting complex in photosystem II, and it moves from molecule to molecule until it reaches P680, which is in the reaction center complex of PS2. So the second step is that an excited electron from P680 is transferred to the primary electron acceptor. And so this results in P680 turning into P680 plus because it's losing an electron, so it's losing a negative charge, so that's where the plus comes from. So then the third step is the splitting of a water molecule by an enzyme or enzymes, and the electrons from this splitting are transferred from the hydrogen atoms to P680 plus. So it reduces it back to P680, and oxygen is released as a byproduct. All right, so the fourth step is each excited electron passes from the primary electron acceptor of PS2 to PS1 through an electron transport chain. So the exergonic fall, the fifth step is that energy released by the exergonic fall of electrons to a lower energy level drives the creation of a proton gradient across the thylakoid membrane. So this diffusion of protons across the membrane drives ATP synthesis. The sixth step is that in PS1, like in PS2, transferred light energy excites P700. So that causes it to lose an electron. So we now call it P700 plus because it's losing an electron. So P700 plus then accepts an electron passed down from PS2 through the electron transport chain. So the seventh step is that the electrons fall down an electron transport chain from the primary electron acceptor of PS1 to the protein ferredoxin, or FD. And the eighth step is the enzyme NADP plus reductase catalyzes the transfer of electrons from ferredoxin to NAD plus. And so it needs 2 electrons for its reduction to NADPH, which is our final product. And it removes a hydrogen ion from the stroma. All right, so this is basically what I was just talking about, sort of simplified. But the Calvin cycle, which we'll talk about in 8.3, I guess, it supplies the NAD plus and NADP plus, and the ADP to the light reactions. So it supplies the reactants, and then the light reaction occurs, and then the products are ATP and NADPH, which go back into the Calvin cycle. So the Calvin cycle provides the reactants and then lets the light reactions do all the work and then takes the products back. So this is sort of an overview of the steps I just described. So basically, if you start at number 1, the light goes into the pigment molecules, it's transferred between them, and then it goes into P680. And then an electron from P680 gets really excited, so it goes up to the primary electron acceptor. So then the splitting, if you look at number 3, the splitting of the water molecule releases 2 hydrogen ions and some oxygen. And the electrons produced from this go back down to the P680. So the fourth step is the electron transport chain. Don't worry about the PQ and PC things. Those are just what make up the chain. And at this point, this is where ATP is produced. So then the electron travels down the chain, and before that happens, or I guess simultaneously, other light is hitting Photosystem I, and it's hitting the pigment molecules of Photosystem I. And so the same thing sort of happens as it happened with Photosystem II, where the light is transferred between molecules and then goes up to P700, which excites an electron that goes to the primary acceptor for PS1. So then the electron that came from Photosystem II now comes down and goes into P700. All right, the last couple steps. This is another electron transport chain, FD for ferredoxin. It carries the electrons down to NADP plus reductase, and that combines with the 2 electrons and the NADP plus and the proton to create NADPH. All right, so we're almost done. So this is basically just sort of an overview kind of thing. So the energy changes of electrons during linear flow can be represented in a mechanical analogy. So this is putting it in some simpler terms, but basically the photon, I feel like I've explained this like 40 times, but the photon excites the electron and brings it up in photosynthesis